Evan, thank you for sitting down with me today. I'm curious, what do plant synthetic biology and agricultural sustainability have in common?
Yeah, well, first of all, thank you for having me. Plant synthetic biology is the discipline of engineering plants, and it's sort of a new conception of how we can use plant genomes, either through transgenesis or genome engineering, to create new and interesting things with plants. Traditionally, the public, regulators, legislators alike have looked at plant engineering and said that this is bad for our health, bad for the farmer, and bad for the environment. And I think the new paradigm, as we see it in the space of plant synthetic biology, is that we can be using plant engineering to both reduce the emissions and the negative externalities of agriculture, reduce the inputs that a farmer is putting into his or her field, and then make ultimately healthier, more beautiful, and delicious food. And so I think there's incredible overlap, but this is a new and sort of uncharted domain of plant engineering that I explore with my research.
Now, at UC Berkeley, you're developing some new CRISPR-Cas9 gene editing tools that are kind of shaking things up. Can you give us a sneak peek into that?
Yeah, so my work deals in testing new and largely recently discovered CRISPR molecules. So these include some Cas9s and then also all sorts of proteins from the wide world of CRISPR enzymes. Ultimately, all of these have some potential in agriculture, but absent incredible genomic inference and the ability to understand how to use CRISPR, they're essentially useless. So a lot of what my research is about is using genomic tools, functional genomic analyses, basically probing at genomes to understand where we can most effectively edit with CRISPR tools to elicit a certain phenotype. And sometimes these phenotypes are things that we're used to, like improving drought tolerance or creating resistance to some pathogen or pest. But sometimes they're all about producing new phenotypes. So maybe bringing a trait out of a plant that has never existed in nature by way of our genomic sort of prediction ability.
Now, you've said that gene editing tools need to be democratized. What do you mean by that, and why is it important?
Yeah, well, it's no surprise to anyone that's listening to this podcast that GM is traditionally a field that's thought of being under the purview of large companies that are extremely well capitalized. What is a little bit different about CRISPR than previous genetic engineering technologies is that the barrier to entry is very low. It's very inexpensive, it's relatively easy to use, and it has the potential to work in crops where we don't know that much about their genomes or their population structure. And so it stands to reason that CRISPR could be enormously helpful in empowering farmers and breeders and agronomists all over the world to improve the way that they farm. So I think there's a lot of onus on us in the gene editing space to democratize this technology so that it can be accessible and interpretable for scientists and farmers and breeders all across the world. So a lot of my project as a scientist and student has been to disseminate these tools and the knowledge necessary to perform CRISPR so that the broader global community can benefit from its discovery.
And before I let you go, you're a Borlaug Scholar this year. You're here at the NAPB meeting in St. Louis. How does being a Borlaug Scholar and coming to the NAPB meeting help to advance the world of gene editing? Yeah,
well, the conversation I've been having all today is that really breeding is where the rubber meets the road, and the buck stops with breeders. And so we can devise the most brilliant CRISPR edit that we want in the laboratory, but it could live and die at the lab bench. So a lot of my individual purpose in being at this meeting is just to mingle with and sort of become more familiar with the broader breeding community of American plant breeders and understand how my field, the discipline of genome editing and genome engineering, intersects with and sort of overlaps with some of the sort of the more disciplinary breeding faculties. And I think that this is a really new and vibrant space. I think there are some companies that have sort of ventured into this space, but largely we're still sort of negotiating amongst ourselves the role that gene editing will play in the broader project of crop domestication and improvement. And so I'm very pleased and very excited to be here.
Yeah, that's a great meeting. Thank you for your time, sir.
Thank you.